Swayam Prabha. Digital India. Educated India.
Hello everyone, welcome to the course on computer aided drug design. We will continue on the topic of ADME. A is the absorption in the GI, D is the distribution in the plasma and other tissues, M is the metabolism that may be taking place in liver and other parts inside, and E is the excretion. So we said absorption, we looked at the weak acids and weak bases because they are very important. The pH inside the GI region changes tremendously. For example, if you look at pH in the stomach 1.4 to 2.1, okay, then the drug may go to duodenum, then it keeps traveling, okay, jejunum where the pH is 4.4 to 6.6, ileum pH is 6.8 to 8, and then colon 5 to 8. So there is a large variation in the pH starting from very highly acidic to basic. So most acidic drugs and weakly basic drugs are absorbed in the stomach. Basic drugs are ionized in stomach and ionized in intestine, so they are absorbed in the intestine. So you need to design depending upon where you want the drug to get absorbed, and especially if it is a fast acting or quick acting, you want it to get absorbed here, slow acting you want it to get absorbed down the line, and so on actually. So we introduced this particular equation for pKa related to pH, pKb related to pH, log of charged versus uncharged. So if you have charged molecule, they are highly polar, they will not cross the membrane barrier. So ideally it should be in the uncharged form so that they can cross the lipid membrane barrier. More the charged it will be, it will not cross. So that is the entire strategy of absorption. Distribution, we said the body is like a big vessel, like a big pot containing a lot of water. So the drug gets distributed, and of course it is not uniform because you have plasma, we have the other tissues, so many different there, intestinal fluids, fat tissues, intracellular fluids, transcellular fluids. So some drugs will get distributed only in the plasma. If it is a highly lipophilic, it may get absorbed in the tissue. So the volume of distribution may change depending upon the type of drug, and as I showed that the maximum concentration of that drug in the plasma will depend upon the volume of distribution. Okay, higher the volume of distribution, lower will be that max concentration, lower the volume of distribution, higher will be that concentration. So this equation, volume is equal to dose by C naught, dose is the amount of drug we give. So they are inversely related, the volume and the maximum concentration it reaches. Depending upon the type of drug these will change, and depending upon so many parameters the V can change. Then I also showed you a very interesting picture, this was taken from this reference. Warfarin for example has a very low volume of distribution, whereas if you take a chloroquine which is antimalarial, warfarin is a blood thinning. So chloroquine has a very high volume of distribution. Okay, so many drugs and alcohols for example, okay, also have different volumes of distribution. So as the volume of distribution becomes higher and higher, concentration also keeps going down and down actually. Okay. Then came metabolism, because there is a lot of biotransformation taking place inside the body, especially in the liver and other places, because of presence of a lot of enzymes, oxidoreductase type of enzymes, lipases, esterases, hydrolases, and there are two stages of biotransformation. Stage 1 where either hydroxylation or oxidation reduction takes place, and then the second stage there are things like sulfate type of groups get added. Okay. So the drug action may increase or decrease okay. So, the drug may be more potent or less potent, you may be forming metabolites which may be toxic, okay. So, all these issues can happen. So, phase 1 changes drugs and creates for site for phase 2, okay. Oxidation, reduction, hydrolysis, these are the 3 things that happen. In phase 2 what we have is it couples groups to existing in phase 1. So, conjugation for glucuronide with glucuronic acid or sulfate and so on, actually this is phase 2. So, the compound becomes hydrophilic, so it gets excreted in the urine. In fact, the metabolism is supposed to be good so that the body is cleaned with the toxins, but drug also is viewed as a toxin and gets metabolized. Many factors affect this biotransformation: race, okay, age, the sex, the species. Monkeys may have different metabolism as against horses or animal like rabbits or human, okay. Clinical or physiological condition, sick patient, healthy patient, if we are giving other drugs, okay, those drugs may be acting on these drug and they may getting metabolized. Food, what type of food we are eating, for example charcoal grill, grapefruit juice, okay. So, it enhances some cytochrome P1A, charcoal grill gets, okay, grapefruit juice CYP, cytochrome P3A. So, the metabolism will get affected depending upon the type of food. First pass metabolism, that means when it goes through the liver as a first pass, you see a lot of metabolism, and after that it reaches a steady state. Okay. So, absorption, distribution, metabolism, excretion is the fourth. You have glomerular filtration rate, GFR, glomerular flow rate of filtered fluid through the kidney, okay. So, this is called GFR. Creatinine clearance rate, that is CCR or CL, is the volume of blood plasma that is cleared of creatinine per unit time. This is a very useful measure of understanding GMR, okay. So, how much of creatinine gets removed per time from the blood plasma. So, that tells you this GFR. So, you have the drugs, we have oxidation or conjugation. So, you may find metabolites, stable adducts. So, if it is polar, immediately it goes through the renal urine. If it is non-polar, these are stable adducts, it may go into the biliary elimination stools. So, this is how drugs get excreted from the body. So, we have oxidation, conjugation, some stable products, metabolites, because these are all polar, it gets removed in urine, non-polar it goes, it will get removed in stools. So, generally urine is the main. The GFR, glomerular filtration, removes all drugs below 25,000, and as you know most of our drugs are around molecular weight of 500 Dalton or less. So, okay, so chances are drugs can nicely get excreted through urine. Reduced by plasma protein binding, only a portion of plasma is filtered. Tubular secretion active carriers process for cations and for anions, and inhibited by probenicide. There is a passive reabsorption. So, as it tries to come out, there could be again reabsorbed of lipid soluble drugs back into the body across these tubule cells, okay. So, it gets excreted and there is a passive reabsorption of some of the lipids molecule, okay. Effect of pH to make more weak acid drug present in ionized form in alkaline pH, therefore, it is less reabsorbed and excreted faster. So, if you have more weak acid drug present in ionized form in alkaline pH, chances are it will not get reabsorbed. So, they will excrete faster, and same thing vice versa for weak bases. So, if you have weak bases, chances are it can get reabsorbed. Okay, so you have something called pharmacokinetics, which studies this entire ADME in a quantitative manner. So, what do? In human we look at the blood, we look at the urine, we look at fecus, we look at expired air, and then we find out concentration of the drug, okay, and then we see how the drug gets removed from the body. So, concentration in urine into volume of urine per minute divided by plasma concentration, that is called renal clearance. Okay. Concentration in urine, volume, that gives you total amount of drug that is getting removed in urine, divided by plasma concentration, that is renal clearance. If neither secreted nor reabsorbed, then clearance is equal to clearance of inulin, that which is 120 ml per minute, because inulin is neither reabsorbed nor secreted by the kidney after glomerular filtration. So, its rate of excretion is directly proportional to the rate of filtration of water and solutes across the glomerular filter. So, we can use inulin to check how good the system is in excreting it. If completely cleared by secretion, then clearance equal to clearance of P hippuric acid, that is renal blood flow, which is around 700 ml per minute. Okay. So, these parameters are very useful to understand the system, how the urine, the kidney works, and how the clearance in the urine happens, and it also gives you a measure of GFR. Okay. So, if you monitor the drug in the plasma, if it is given as an oral drug, the drug concentration increases, the x axis is your time, y axis is your concentration. So, it reaches a max and then starts coming down because of excretion. So, we can write a first order relationship, Ct concentration is equal to C naught, that is the maximum it reaches. Okay. So, we can, if we extend it, the C naught e power minus t k L, k elimination, that is called, that is rate constant of k elimination. If you take ln of both, you are going to end up with the ln k elimination. If you take logarithm, this is with base 10, this is with base e, we end up dividing by 2.303. Okay. So, what do? We extend this right up to the end. If you have the x axis as time, y axis as logarithm C naught rather than ln, okay, then we draw this line, extend it and see where it touches, and you take anti-log, that will be your C naught, okay, and then the slope will be your k elimination. So, we get both the C naught and k elimination. So, this is a very useful relationship to understand how the drug will concentration falls down with plasma. We can even calculate what will be the half life of the drug, that means the concentration comes to half its max concentration, time it takes for the concentration to reach that we can find out. Okay. So, these are very useful pharmacokinetic parameters. In addition, we can calculate volume of distribution, dose by C naught, that is C naught is this, we know how much dose we gave, plasma clearance, k elimination is nothing but the slope of this line into volume, that is plasma clearance, plasma half life, that is how long it remains, T half is equal to 0.693 by k elimination. Bioavailability is the area under the curve, okay, when if it is given as oral divided by area under the curve when it is given as a IV, that gives you the bioavailability. So, from this graph we can do a lot of calculation. So, we extend this and then from there take anti-log, we get C naught, this slope will give you k elimination, volume of distribution is dose by C naught, plasma clearance is Cl is equal to k elimination multiplied by volume, which you get from here, T half plasma half life time 0.693 by k elimination. If you have this similar graph when it is given in IV form, area under the curve in oral divided by area under the curve in IV form. Okay. Like I said, generally bioavailability has to be very high, so that the concentration at the target site is good, the efficacy of the drug is good, we do not have to give too much dose of drug, so possibly toxicity also can be lower. So, if you have multiple doses, so there is going to be some accumulation of this drug maybe in the tissues, or it is not fully removed from the plasma. So, you may have this type of up and down, this is loading doses, that is why sometimes the first dose or second dose may not be effective, but third, fourth doses become effective because there is a slow build up of the drug because of maybe tissue absorption, then further desorption and so on actually. Okay. Of course, you need to watch out if you know the toxic limit of the drug, you work within the toxic below the toxic limit. So, if you have the effective range and toxic, this will be nice to operate within, which effective could be if you are looking at antibacterial, minimum inhibitory concentration. So, you want the drug all the time to be above that MIC, but it should be less than the toxic limits. Okay, so to sum up, ADME, we have the dose given orally, you have the stomach, pH is 2, we are talking about stability at these conditions, and then there are enzymes which may degrade the drug. Then solubility, the drug has to be soluble, pH over a wide range. Intestine, pH has become bigger, it has to be still stable. Then of course you are forming metabolites, they have to be stable. Then we have the permeability, that is, it is crossing the barrier. So permeability, we talked quite a lot about permeability, what is important in log P. Generally it is passive type of permeability, just like food, pH is big. We have something called PGB efflux, okay, I will talk about this later, the PGB efflux. There are P glycoprotein which will throw the drug out, okay. So those things we will talk about it. Then of course the metabolism, phase 1 metabolism, phase 2 metabolism, and then you could also have proteins binding. There are, if you have acidic drugs, protein binding could be a big problem, okay. Then apart from liver, you need to consider other enzymes present in the body, whether your drug gets stable or not. And then of course the kidney, the urine excretion comes into the kidney, permeability, all those come into the value. And then permeability to the specific site, again through passive, you can have again PGB efflux coming into, that means P glycoproteins may be throwing your drug out, and that will lead to reduction in its concentration. And then this distribution, volume of distribution which we talked about, which can alter the concentration of the drug in the body. And then again we have the permeability, then cellular partitioning, if the drug is going in the presence of cells, there could be partitioning of the drug, metabolism happening, and finally reaching target site. So lot of steps before which the drug reaches the target sites and exhibits bioactivity. Okay, so the bioactivity is not disturbed, but the bioavailability and stability, solubility, and all these factors are disturbed because of presence of all these environmental changes, as well as the presence of enzymes and so on. The activity does not get modified, efficacy can get modified because the drug can become less efficacy because of the metabolism of the drug, formation of new molecules and so on actually, okay. So this entire set of steps affect the drug action overall. That is why many drugs, when they go into clinical trials, whether it is animal or human, fail, although it may have shown very good activity in the lab, in enzyme assays or some bacterial assays and so on actually. Okay. There are many rules, okay, many models, rules which give a feel of whether a drug can be bioavailable, orally bioavailable, whether it will satisfy all these conditions like good solubility and good GI penetration and so on, okay. There are many rules. So we can have a look at some of these rules, and when you have a new compound designed, you can check it out whether it will satisfy all these rules or whether it does not satisfy all the rules. If it does not satisfy, and if you feel there could be some problem, you need not take that particular compound for activity selection. So we can use these rules also for screening a large number of molecules, okay. That is what we do. Okay, the very well studied, well understood, well considered is called the Lipinski's rule of 5. Okay. Lipinski and his co-workers at Pfizer looked at a large number of drugs, that is drugs which have sort of accepted by FDA and passed the FDA. So they picked up and they said all these drugs satisfy certain conditions. The first one is molecular weight less than 500. So they said you should have molecules which are reasonably small, less than 500, you cannot have very large molecule. Please note this is for oral, this rule is, all the rules I am going to talk about is for oral drug, not for IV or IP and so on. Number of hydrogen bond acceptors less than 10, that is oxygen and nitrogen, okay, are defined as hydrogen bond acceptors. Please note if you have an acidic O minus, then it will not be an acceptor, but oxygen and nitrogen are defined as hydrogen bond acceptors. Hydrogen bond donors, that is NH or OH, should be less than 5. These are very important because these determine the hydrophilic lipophilic nature of the molecule, plus also how they go and bind and with various enzymes. So number of acceptors should be less than 10, number of hydrogen bond donors should be less than 5, and log P which I defined, that is the octanol water partition coefficient, should be less than 5. That means if it is more than 5, you are talking about a molecule which is very hydrophobic, and if it is less than 0 or less, then we are talking about a molecule which is very hydrophilic. So both we do not want. So according to them it should be in this region, but there are rules which narrows this region also. So according to them, if 2 parameters are out of range, then chances are it will definitely have a poor absorption or permeability, and bioavailability also will be low. So do not take up that molecule. If one of the parameters are bad, chances are maybe it may have reasonably good GI absorption, okay, but if 2 do not. Okay, so that is the rule. So molecular weight less than 500, the number of hydrogen bond acceptors less than 10, number of hydrogen bond donors less than 5, log P less than 5, that is 0 to 5. This is called the Lipinski's rule of 5. Similarly, there are many rules, as I said, we are going to look at some of them. We have another rule called Gose et al. This is called drug likeness property. Okay. So they came up with the law, again log P. There are many prefixes M, A, so these are calculated using different softwares. So they gave little more range, minus 0.4 to 0.6. Molar refractivity between 40 to 130. So they brought in a new parameter. This is the volume occupied by an atom or group and is dependent on the temperature, the index of refraction and the pressure. Okay, this is the volume occupied. So they bring in the volume between 40 to 130. Molecular weight, look, Lipinski said less than 500, whereas they are bringing 160 to 480. They also bring number of atoms 20 to 70. So they have made some changes. They came up with new set of parameters or descriptors, we call it parameters, descriptors, features of the drug. This is called Gose et al. Drug likeness property. Then there is a Weber rule. Weber rule for good oral bioavailability in rats. They said less than or equal to 10 rotatable bonds. Why rotatable bonds are important? Because if we have more rotatable bonds, it can take up different conformations, so the binding with proteins have large degrees of freedom. The polar surface area should be less than 140, okay, angstrom square. If it is more, then of course it becomes more hydrophilic, it will not permeate through the GI. Less than 12 hydrogen bond acceptors plus donors, okay. So they did not differentiate between hydrogen bond acceptors separately, donors separately like Lipinski's, okay. This is called Weber rule. Then comes another rule, that is Muget et al. Rule. So what they did was they looked at a molecule and saw whether it had these type of functional groups, amine, amide, alcohol, ketone, sulfone, like that, you know. So they gave a score based on the presence of a structural fragment typically found in drugs. So they say drugs will have only these groups: ester, urea, amidine, guanidine, carbamate, carboxylic acid, sulfonamide, sulfone, ketone, alcohol, amide, amine. So you give 1 mark for that, give 1 point for each non-overlapping pharmacophoric feature. That means if it has got 2 ketone groups, do not give 2 marks, just give 1. So molecules with a score of 2 to 7 are classified as drugs. So look at your molecule and see how many these groups are present. So you give 1 mark for each of this non-overlapping, and if it falls between 2 and 7, then you classify as drugs. And these groups, even if it is present 1, compounds containing a single feature of carboxylic acid or amine, then you can classify it as a drug, okay, even if it contains single feature that is there. So how did they do it? They looked at the various functional groups present in all the commercial drugs approved in FDA, oral drugs, and then they said we will give 1 point for presence of each of these non-overlapping group, and if the score is between 2 to 7, you can classify it as a drug, and if the compounds contain this single, even if it contains 1 single feature, then we can also classify it as a drug. That is called Muegge rule. And the effective permeability through the intestine is calculated from this equation: log P effective, PSA is polar surface area, HBD is hydrogen bond donors. Hydrogen bond donors means OH or NH. So if this figure was taken from this reference, as you can see, okay, as the polar surface area is low, that means it is lipophilic. The absorption fractional, the absorption in the intestine is very, very high, and as the polar surface increases, that means it becomes more hydrophilic, then it dramatically falls down and it comes to very low value when the area polar surface area is above 140 angstrom square. That is why, as you can see in one rule, we said it should be less than 140 angstrom square, but ideally it should be less than 100, so that you have absorption at least 50%. Okay. So polar surface area should lie in this region, that means lipophilicity is good, but of course we do not know whether solubility will be good if you have more lipophilic. So you want to work in this region, at the same time have a soluble molecule, you do not want to work in this region. So that is the based on polar surface area and hydrogen bond donors, trying to look at how the effective passive permeability will be through. Then came log P. As I said, log P plays a very important role. Log P is octanol water partition coefficient. It gives you hydrophobic hydrophilic balance. Hydrophilic means soluble in water and blood, hydrophobic means soluble in the lipid, and it gets transported across, and most of the drugs have log P between 2 to 4. 4 or 5 highly hydrophobic. You have some anti fungal drugs having very high log P. If the log P is very low, it is highly hydrophilic, it might not cross the barrier, so it may be given in the form of IV. Look at this, morphine, very low log P, highly hydrophilic. You have so many OH groups here, okay, nitrogen, so it is always given as IV. As I said, C or M, they are all using different softwares actually. Cocaine, okay, 2.72, Indenivir 2.78, imipremine, high log P, because it has got only 2 hydrogen bond acceptors, otherwise lot of CH2s, highly hydrophobic molecule here. Okay. Although this has got many O's, it also has got many CH2 groups here, the benzene rings and so on actually. Okay. That is why log P is not very bad. The biopharmaceutical has classified drugs into 4 categories. Okay. It is called highly soluble, highly permeable, then we have poorly soluble, highly permeable, highly soluble, poorly permeable, poorly soluble, poorly permeable. So 4 classification. Okay. The highly soluble, highly permeable, there is no problem, drugs will nicely get solubilized in GI and they will also nicely pass through the membrane. Okay. For example, look at this diltiazem, this is heart pain, high blood pressure, cardiac arrhythmia and so on. So, it has got highly soluble, highly permeable. So, it has got a lot of hydrogen bond acceptors are there, at the same time it has got a lot of CH2. So, it is nicely balanced. Enalapril, this is for hypertension, this is also highly soluble, highly permeable. Labetalol is chronic and acute hypertension, these are all salts as you can see here, okay, salts. Propranolol, hypertension and chronic heart failure. So, these are coming under class 1 highly soluble and highly permeable drug. Now, let us look at class 2 type of drugs, low solubility highly permeable, that means its hydrophilicity is less, permeability is good, that means it is hydrophobic. Okay. Fluorbiprofen, just like ibuprofen, okay, fluorbiprofen relieves pain, tenderness, swelling, stiffness, given for osteoarthritis, rheumatoid arthritis. Naproxen relieves pain, tenderness, swelling. So, it has got low solubility, that means a lot of drug can get excreted from the body because it does not fully dissolve, but permeability there is no point, no problem at all. Highly soluble but permeability is low, that means it is hydrophilic type of drug, so it does not cross the GI. Acyclovir, this is for herpes, so you will see a lot of nitrogens and oxygens. Famotidine, peptic ulcer, gastroesophageal reflux, that means acids come out through the esophagus. Look at this, a lot of nitrogen, so it is quite hydrophilic. Nadolol for high blood pressure, migraine, headaches, chest pain and so on, again it has got a lot of oxygens. So, high solubility it does not permeate through that. So, obviously we need some carrier to take it inside the body, okay. Whereas in the class 2 solubility is low, so we can make salts, in fact they have made salts of this, okay, they are making salts of this, but still solubility is low. So, we need to increase that, so that it dissolves better. Here solubility is good, permeability is poor, so we need some carrier type of system to carry it across the GI. Next one, low solubility, low permeability, everything is bad, it does not dissolve properly or nicely in the GI, at the same time permeability is also very poor. Terfenadine is for allergic, furosemide reduces swelling, fluid retention caused by various medical problems. So, these drugs obviously have serious problem, we may have to make if these drugs are given in large doses you can have toxicities also. So, there has to be some structural modifications to these drugs to improve their solubility as well as permeability. Okay. Solubility we can improve easily making salts out of this drug, so that or putting in OH type of groups, acidic groups, but permeability may go down. If you want to improve permeability, we block OH or NH with CH3, that means make it more hydrophobic. So, these are the 4 different types of classifications, it is called biopharmaceutical classifications: high soluble, high permeable, low soluble, high permeable, high soluble, low permeable, low soluble and low permeable. Okay. So, for each of these drugs we may require different strategies for improving either the solubility or the permeability. So, we will continue further on the bioavailability and ADME. Thank you very much for your time.